Next lesson in this recording, we're going to talk about cardiac muscle tissue, cardiomyocytes, and a little bit about the potential electrical activity of the heart. So as we look at a cardiomyocyte, cardio is a root word that means muscle. Excuse me, no, I'm wrong there. Cardio is a root word that means heart. Myo is a root word that means muscle. Cyte meaning cell. Cardiomyocyte means heart, muscle, cell, literally when you look at the root words. And as we look at a cardiomyocyte, they're fairly short cells that are striated. So you can see there's some striations here in this photomicrograph. And they're also branched cells. So there's a branching appearance or structure to the cells as we look at them within our photomicrograph. So there's some branching that has occurred with our cardiomyocytes. And something that's important here is that these cardiomyocytes have a very, very poor regeneration, right? If there's damage to the cardiomyocyte, there's very little regeneration. It's almost entirely going to be fibrosis or scarring that occurs. So once somebody has a heart attack or a partial heart attack, they're going to have dramatically decreased cardiac function as a result. They're never really going to reclaim that prior function. Cardiomyocytes are connected to an adjacent cardiomyocyte with an intercalated disc. And we talked about these very briefly in AMP1. Let's look at these intercalated discs. I'm circling some in yellow right now. These intercalated discs have a very special cell-to-cell junction called a gap junction. That's really the one that I need to emphasize to you. This gap junction is a little hole that serves to allow for the electricity to flow from one cell to another cell. And as we're looking at this, it allows for the cytoplasm of one cell to flow adjacently into the other cell. They are also going to have what are called interdigitating folds. When we think of digit, think fingers. And then when we think inter, think in between. So if we were to zoom in really close on the intercalated discs, these interdigitating folds are just little fingers that fold back and forth to connect cell A to cell B. And then we have the gap junctions in between them. And those gap junctions allow for ions to flow from one cell into the other cell. And if we allow electrically charged particles or ions to move from one cell to another cell, we allow for electrical activity to flow directly from one cell to the other cell. In short, these intercalated discs allow for a continuous transference of an action potential. They are an electrical synapse. So this figure illustrates this idea better for us. The mechanical junctions in particular are going to anchor the cardiomyocytes together. One cardiomyocyte anchored to the adjacent cardiomyocyte. There's also going to be what is known as the fascia adherens. And this is a great word here, adherens. It adheres. And when we think of fascia, think of extracellular connective tissue. So we have the fascia adherens within our intercalated discs. And then we also are going to have desmosomes. These desmosomes, shown here, are going to allow for a flexible connection between the cardiomyocytes. And flexible connections generally are going to be more resilient than rigid connections. And these desmosomes do a great job of keeping the cardiomyocytes from being pulled apart. And then we also had those gap junctions, which serve as an electrical junction or electrical synapse. This means that when we look at the myocardium of either of the two atria or the two ventricles, they act like a single cell because ions and cytoplasmic contents will flow from one cell into the other cell back and forth. Now, as we look at cardiomyocytes, we need to really emphasize here that they depend almost exclusively on aerobic respiration to make their ATP. They need tons and tons of ATP, so they're going to very extensively rely on aerobic respiration. Anaerobic respiration just doesn't do it for these cells. They have huge energy needs. They're also going to have lots of myoglobin within the cardiomyocytes. Recall from last semester, myoglobin is a oxygen storage molecule within a muscle cell, and cardiomyocytes also have a lot of glycogen or complex carbohydrates stored within them. So in addition to storing oxygen, they also store sugar. And cardiomyocytes have very large mitochondria. They are going to be approximately 1/4 mitochondria by volume. So they have lots of they store oxygen, they store sugar, and they have lots of ATP factories within them. They are ideally situated to pump out lots of ATP. Cardiomyocytes can also use different organic fuels. They are able to use fatty acids. They are also capable of using glucose, ketones as a protein byproduct, lactate, and or amino acids. These cardiomyocytes are more vulnerable to oxygen deficiency than a lack of a specific fuel. So thankfully, when we think of like our brain is almost exclusively dependent upon glucose for energy. Our cardiomyocytes, our heart, can use fat, glucose, protein, or lipids, glucose, carbs, or proteins to make energy. It's really the oxygen deficit that's the problem for our cardiomyocytes. Or the cardiomyocytes limiting factor typically is oxygen, not fuel sources. Cardiomyocytes are very fatigue resistant because they make very little use of anaerobic fermentation. Part of that is because they store so much oxygen and extra fuel within their cells. And think about this. I mean, if your heart was prone to being fatigued, it means that your heart would be unable to pump blood for your entire life. So we need these tissues to be very fatigue resistant. Now, as we look at the electrical conduction system of the heart, we're going to camp on this slide for a little bit here. This conduction system is going to serve to coordinate our heartbeat. We're going to have an internal pacemaker. We're going to have some electrical pathways. And these systems serve to generate and then transmit electrical impulses. We're going to start off with the SA, or sinoatrial node. This is going to be made of some modified cardiomyocytes, which makes it rather difficult to see in a heart dissection because the cardiomyocytes that make up the SA node blend in with the other cardiomyocytes that make up the wall of the right atria. But it's located right here. There's our SA node right here. And when we look at this SA node, or the pacemaker node, it will spontaneously depolarize, typically with the highest frequency of any tissue in the heart. There are lots of leak channels in those cells that cause them to periodically and rhythmically spontaneously depolarize so that we have a baseline heartbeat. And as we look at this signal, once the SA node fires, the action potential will travel all the way across the atria. They'll go through the right atria and all the way over here to the left atria. And that's the signal spreading throughout the atria. So both of the atria contract at the same time. Let me back up a slide here for us. I want to talk about this while there's a picture. So after our atria contract, that signal from the SA node also is going to travel to the AV node. Sometimes, and that AV node, and I like that, it's the atrioventricular node. It really does a good job of telling you where it is. The signal pauses for a fraction of a second at the AV node. And then from there, that signal will travel down the pathway, down the AV bundle, and then we're going to have the AV bundle branches. The old school name for this is HIS. So we'd have HIS, HIS. So we'd have traveling down the HIS branch, the HIS bundle to the HIS branches. And then that signal is going to get all the way to the apex of the heart and continue traveling through the branch fibers. As those bundle branches move up, they start to terminate with very fine little branches. And those very fine little branches are referred to as the Purkinje fibers. It's kind of a fun word to say, Purkinje. Ultimately, what we end up have happening is first the two atria contract at the same time, then we skip half a second, and then the two ventricles contract at the same time. So that AV node typically is going to be the gateway to the ventricles. The electrical signal that goes to the ventricles has to make it through the AV node first, and there's going to be a fibrous skeleton that we talked about in just a couple recordings ago that helps to electrically insulate the atria from the ventricles so that we can only have the signal get to the ventricles through that AV node. The AV bundle will branch to the left and right bundle branches, and then ultimately those bundle branches will branch into Purkinje fibers. The new school name we're looking at now is subendothelial conducting network, which I guess is the more descriptive name, but I guess I'm a little crusty. I just love the word Purkinje. It's fun to say, Purkinje fibers. And then we'll have the two atria depolarize and contract. Now, as we're looking at controlling the heart rate with our sympathetic versus parasympathetic nervous systems, we find that the sympathetic nervous system is going to upregulate. It increases our heart rate, and it also is going to increase contraction rate. When we think of the parasympathetic system, nervous system, it slows down our heart rate. So these two will work back and forth. Now, if we remove all sympathetic and parasympathetic influences on the heart, the SA node, the pacemaker of the heart, is still going to have a baseline heart rate. But these two branches of our autonomic nervous system, the sympathetic and parasympathetic branches, can upregulate can excite or inhibit the pacemaker cells of the SA node. When we have a contraction of a chamber of the heart, we refer to that as systole. Systole means contraction of a chamber of the heart. So if we have atrial systole, the atria are contracting. Conversely, we have dystole. Dystole is typically going to be relaxation, so we can have ventricular dystole, relaxation of the ventricle. And while systole and dystole, or diastole as some people pronounce it, typically can literally mean any chamber of the heart. Pragmatically speaking, all we care about is the ventricles, because when that left ventricle contracts or relaxes, that determines our systolic or diastolic blood pressure. We also are going to have what is known as the sinus rhythm of the heart. That's that baseline heart rate triggered by the SA node. So when you're just chilling, relaxing, you aren't really excited, you aren't having strong parasympathetic or sympathetic influences, your baseline heart rate is typically going to be about 70 to 80 beats per minute. And that's going to be influenced very, very heavily by that SA node. As we look at the pacemaker cells of the heart, that SA node doesn't have a stable resting membrane potential. And this is very key to the function of this structure. Because the resting membrane potential is not stable, there's going to be sodium ions slowly rushing in and raising the voltage of the inside of the cell to threshold. And once we reach threshold, calcium and this is a key distinction here, calcium and sodium voltage-gated channels pop open, and those ions rush in. And because we're letting both calcium and sodium ions rush in, we're going to have an incredibly fast depolarization event. Typically, that depolarization event for these pacemaker cells, and if we look at the numbers here, these are not the same numbers you had when we worked with when we worked with neurons in our nervous system last semester. So we have -60, -40, and typically 0 here, 0 millivolts-ish as our peak voltage. So those numbers are going to be a little bit different. And as we're looking at this depolarization event peaks at about 0 millivolts. At 0 millivolts, the calcium and sodium voltage-gated channels close. The potassium voltage-gated channels pop open. Potassium flows out. There's efflux of potassium. And then as we lose positive potassium ions from the inside of the cell becomes increasingly negative. And typically at about -55, -60-ish millivolts, the calcium voltage-gated channels close. And then we're going to start the process all over again. And just like with the nervous system, when we talked about action potentials, there are sodium-potassium pumps. And they aren't shown on this figure, but those sodium-potassium pumps get the sodium and potassium ions back to their initial ion concentrations to reset the ion concentrations. So as we look at this SA node, once it fires, right here, that determines ultimately an entire series of events that result in the atria contracting, then the ventricles contracting, and blood leaving the heart. As we look at the timestamps here, I mean, this is cool. Look at this time here. So we have a depolarization event right here. If we start at time 0, we're starting at the bottom of resting. And from time 0 to the time it takes us to get back there, we go from timestamp 0 to timestamp 0.8 seconds. So this is for a quote-unquote average adult. We're looking at about 0.8 of a second between action potentials being triggered by our pacemaker. This gives us a resting heart rate of 75 beats per minute on average. That's all we have for this recording on the conduction system of the heart. If you have any questions, comments, or concerns, please feel free to post them on the class discussion board or to shoot me an email. And as always, happy studies.